All right, let's move on to thiols and sulfides. These are essentially the sulfur analogs to alcohols and ethers, and there are a lot of similarities, but some key differences. Thiols themselves are the sulfur analog of alcohols. Thiols are also known as mercaptans because mercury has an unusual affinity to sulfur. If we have a sulfur compound, it can attract mercury, so mercaptan means mercury capturing. So to name thiols, right, to name a compound that's RSH, that is a thiol, we can name it with a thiol suffix, just like naming an alcohol normally. If we have to have a prefix to it, we can use mercapto as a prefix, just like we would do hydroxy for an alcohol. So 2 carbons and an SH is ethane thiol. Ethanol is only called that. It's not called ethanol because we avoided the double vowel. So here we keep the final E in because our ending starts with a consonant. So this is ethane thiol. Cyclohexane thiol compared to cyclohexanol. This is meta-mercaptobenzoic acid. Compare that with meta-hydroxybenzoic acid. So anything you can do with hydroxy, you do with mercapto. Anything you would do just ending a name with ol, you can do with thiol at the end. Just so we're clear about naming precedent prefixes here, let's make this one a little more complicated. Let's, in the cyclohexane thiol, I put a double bond and a methyl group because the thiol will still remain the dominant functional group here, right? The name of the compound will still end with thiol. It'll have a cyclohexene in it though. But because the name ends with thiol, that's where our numbering will begin. Just like how alcohols will begin their numbering, irrespective of where the double bond might be. So this would be, for instance, 3-methyl, 2-cyclohexene thiol, right? And it would be a little better even if we called it cyclohexene 1-thiol. Okay? So 3-methyl, 2-cyclohexene 1-thiol. If we wanted to do the new type of name, it would be 3-methyl cyclohex 2-ene 1-thiol, right? So, well, either way is fine. Thiols are pretty interesting because they tend to smell just terrible. They have awful smells. They're some of the worst smelling compounds that there is. And nature has taken advantage of this. If you take a look at this 4-carbon thiol, this is 2 butene 1-thiol. That's a compound that is present in skunk spray. It's detectable by human noses down to 10 parts per billion. Okay? So tremendously low concentrated needed to be detectable by human noses. Ethane thiol is, I believe, one of the thiols that's used to give natural gas its odor. Okay? Methane itself has no smell, so they add a thiol to it, which can be detected at very low concentrations, just like skunk spray, so you can detect if there's a gas leak. Let's prepare some thiols. Very simple to do by an SN2 reaction with SH-, the hydrosulfide ion. It is an excellent nucleophile, okay, but very weak base. So here's one key difference between thiols and alcohols. The SH- ion is a great nucleophile. It's a very poor base. OH- is a good nucleophile and a good base. The reason for that is that H2S is a much stronger acid than H2O is. So SH- is able to stabilize its negative charge more, but because it's large and polarizable, it's still an excellent nucleophile. So we can do an SN2 reaction. If I have SH-, that can displace something like a primary bromide to give this 1-octane thiol here, an SH. We do have to run this at a very large excess of SH- because we don't want this product to be competing with SH- as a nucleophile. Okay? Even this neutral SH is a very strong nucleophile. So it turns out if we have a thiol, it can be oxidized to form a disulfide. It's the sulfur equivalent of a peroxide. The thing is, disulfides are actually quite stable, where peroxides are very unstable, very reactive. So disulfide bridges, where we have an SS bond in there, are very important structural elements of proteins because there's an amino acid cysteine that has an SH in it, and oxidatively, cysteine amino acids in different parts of the protein can link together, and that helps proteins form into the, helps them fold into their correct shape. So it's an important structural element in proteins. So there's an oxidizing and reducing process that can happen between those forms. Perms for hair have an oxidizing agent in there that links SH thiol groups in proteins in your hair together so that they hold their shape a little longer. So thiols were SH. Sulfides are like the sulfur version of ethers. So we name them with the sulfide ending or with alkyl thio as a prefix. So sulfide instead of ether, or alkyl thio instead of alkoxy. Okay? So simple, especially symmetrical things like this, dimethyl sulfide. Here is methyl phenyl sulfide. Here's one where we have to name it as alkyl thio. Since that's a substituent, if I have something like a double bond, the name ends in -ene, so I use the double bond in that case as the numbering. So 3-methylthiol cyclohexene. So to form a sulfide, we would do the equivalent of a Williamson ether synthesis. Okay? If I start with a thiol, I can deprotonate it and get an S-. Right? It's called a thiolate. Thiolate is a tremendous nucleophile that can displace a primary alkyl halide like that, and we get a sulfide. Right? Just like the Williamson ether synthesis. It so happens that the thiolate ions are just a ridiculously strong nucleophile. Okay? SH- or RS- like that are among the strongest nucleophiles known. So they're so strong that even the neutral product, even the sulfide product, is a very strong nucleophile itself. So if I have a dimethyl sulfide in a favorable enough concentration with something with a leaving group, the neutral S itself can attack a carbon, can attack an electrophilic carbon and displace it to get the sulfonium ion. So that IUM ending is characteristic of positive charges like oxonium, right? There's an O+. This is a sulfonium, trimethyl sulfonium iodide. So neutral sulfurs are really very good nucleophiles as well. So sulfonium salts, it turns out, are powerful alkylating agents. Right? If I have an S- with 3 R groups to it, it can impart one of those R groups onto something else. It can deliver an alkyl group to something else. So, for instance, in biology, norepinephrine, here's norepinephrine, it's alkylated to make epinephrine. The only difference between these is that norepinephrine is just an NH2 on the end. Epinephrine, or adrenaline, is NH2 plus CH3. Okay? It's a positive nitrogen. So what happens is that this norepinephrine attacks, right, the lone pairs of nitrogen attack a thiolate ion biologically. It's part of an important biological molecule. It has a CH3. Here. Let's draw that a little better. Okay, that puts electrons back down onto sulfur there. What we end up is the alkylated norepinephrine, which is regular epinephrine just plus the normal neutral sulfide. So as a way of delivering a methyl group or an alkyl group onto something, anything with a pair of electrons can attack that methyl group, which pushes the electrons back down onto sulfur. So that's very useful key part of biological signal transduction. This can also be very harmful. Look at this structure here. Cl, CH2, CH2, S, CH2, CH2, Cl. This is mustard gas. Okay. Very powerful, very toxic agent used during World War II, for instance. What happens is the sulfur in here can displace one of these chlorines intramolecularly. So what we end up with is this kind of structure. It has a 3-membered ring in it with an S+, and then up, down, Cl. So that's actually present in some quantity in any sample of mustard gas, so that anything biological that has like a nitrogen in it, just like that, will instantly be alkylated. And that's, this is most effective, for instance, in DNA. Here, if I just make a pyridine here, just to represent part of a nitrogen base in DNA, it can attack one of these carbons of the 3-membered ring. What we end up with is alkylated DNA, which is not a good thing. Cl. There, I'll put some extra bonds there. And the problem here is that we still have an S CH2 CH2 Cl. So once this DNA is alkylated, it can make this 3-membered ring again, and another strand of DNA or another part of the DNA can attach to it there, and it prevents the DNA from spiraling together properly or unzipping like it should when it's needing to be replicated. So very powerful agent. Any kind of alkylating agent like that will stop any cell's processes in its track because of this reaction with DNA. Turns out that sulfides can be oxidized fairly readily to sulfoxide. So if I have a sulfide, just RSR, any oxidizing agent like peroxide, H2O2, or bleach, NaOCl, for instance, we can oxidize, or there, we can make a double bond O there, that's a sulfoxide. Notice sulfur has expanded its octet, 2, 4, 6, 8, 10, okay, but still no formal charge. A more powerful oxidizing agent like a peroxy acid here can oxidize it all the way to a sulfone. That's an S with 2 C double bond O's attached. So if we've got some skunk spray, if your dog got hit with a skunk here, let's draw that butene thiol. If I use peroxide, we can oxidize that thiol, right, to like this sulfoxide type derivative here, which is no longer smelly. Okay? Once I've oxidized that sulfur, it doesn't smell any longer. So that's why the sort of accepted skunk treatment is a mixture of a little bit of detergent, some baking soda, peroxide. Sulfoxides are important too. Here's dimethyl sulfoxide. It is an important solvent. It's used quite a bit. It's polar, it's aprotic, so good for SN2 reactions. The dimethyl sulfide itself is an important reducing agent. It can be used instead of zinc, for instance, in ozonolysis reactions, and that's handy because dimethyl sulfoxide is formed as a product, non-hazardous product that's easy to get rid of.